Well, hello, and my name is Milena, and thank you for this opportunity to present our work on immuneML. So this talk will be given by Lonneke and me. The first part, I will try to talk about more technical aspects of building a bioinformatics platform, and then we'll also look into some usability concerns and how to improve the usability of the platform. So the first question when talking about building a bioinformatics platform is, what is it actually? What is a platform actually? Like, is it, what is the difference between the method, a framework, or a platform? If you look through the literature, there is a bunch of papers. Some say, oh, we build this method, some say we build this framework, some say we build this platform, and some even use them interchangeably. So in this presentation, when we're talking about platform, we mean something that is a software that is targeted towards the end user, that is user-friendly and extensive, so that it covers use cases that were the target of the platform a bit more in depth versus a specific methodology. We'll briefly introduce immuneML as a case study when talking about building bioinformatics platforms. And then we'll talk about some technical considerations like the requirements and modular design, agile approach, and so on. And then we'll also cover open source version control, accessibility, different ways to improve usability, documentation tutorials, and of course, if all this doesn't work, what to do with in that case and how to improve the quality of error messages. So just a minor warning, these are our experiences from building a platform and are as such probably subject to change. But so this is basically our experience from developing a bioinformatics platform. So our target use case for this for the immuneML platform was development of diagnostics based on adaptive immune receptors. So adaptive immune receptors are proteins that are located on the surface of immune cells, and they can recognize different pathogens like viruses or bacteria, and then help initiate the immune response that will help destroy those foreign agents in the body. By analyzing these receptors, and there is a collection of all receptors in one person is called a repertoire, we can diagnose an immune-related disease. This is a very interesting and a very hard problem because immune receptors are very diverse. There could be up to 10 to the power of 15 different sequences that we can analyze. But they're also very specific, recognize very specific viruses, for example. So it is very hard to predict recognition. And there are two main use cases in this setup. The first one is predicting the disease state from the immune repertoire, so predicting if a person is sick or healthy. And the second one is, if we look into specific individual receptors, trying to predict if it will bind the target, like a part of the virus. So our goal with immuneML was to develop a platform that will help us to develop and evaluate different machine learning algorithms that are for immune state prediction and receptor binding prediction. So what immuneML does is it takes a data set as input, which is based on this list of receptor sequences, and then employs different data representation techniques and different machine learning approaches in order to build a model that is able to predict the disease. And then in addition, of course, it does some exploratory analysis so that we can learn more about the data set. It can simulate synthetic data, or it can apply the trained models on new data sets. And if you're interested in more details about immuneML, you can see more on our website, immuneML.io.nl. So immuneML will be a running case study. So the first kind of thing that one needs to think about when developing a platform is defining the requirements. So defining the specific use cases that should be covered by the platform. Namely, what type of data will be used, what is the input, what is the output, and what analysis should be performed. So in immuneML case, we defined that we want to work on air data. So those are amino acid sequences or sets of sequences in case of repertoires. And we want to provide that as an input along with analysis specification. For example, saying, oh, we want to train a machine learning model to predict the disease status. The output of such a platform is our trained machine learning models or reports and predictions generated on the given datasets. It is very important to define the requirements early on, because bioinformatics in general is a very complex domain, and it is not really possible to right away come up with everything that you can. There is no universal solution for a bioinformatics platform, so it's very important that you precisely define what needs to be done in the platform, so that even the if you go too broad, like the abstractions that you make could be wrong, the development process could be too complicated, and it doesn't need to be. So one other point that we came across while developing this platform is a so-called agile approach. That means that one good way to develop software is to approach development in iterations, so that at the end of each iteration, we always have a working version. So this working version will have only a subset of features, but will be very useful to learn more about the problem and to learn more about the implementation, so that it can guide the next iteration. For example, in immuneML development cycle, we initially had very long iterations, which significantly delayed the feedback from collaborators regarding the speed, the workflows, the specification, and so on. But once we switched to these faster iterations and asked for feedback more often, we were able to develop the platform much faster. And if iterations, as if I convince you that iterations are good, what that means is that we will end up often doing some sort of refactoring, and that's kind of but that's a good thing, because in that way we steer the platform in the right direction. For refactoring as such to be possible, we need to define good interfaces, and in case of object-oriented design, that also means do proper encapsulation and good separation of concerns. So as to allow for easier refactors. And there will always be refactors regarding if you do agile development or not. Defining, making a good design, making good, clear interfaces is always a plus. For example, we had a repertoire class in immuneML that handles all the sequences and related metadata for one individual. But as the platform progressed and we tried with larger and more realistic repertoires, the problem occurred that we cannot fit many repertoires in the memory anymore, because one repertoire has around 300,000 sequences plus associated metadata. And as the dataset size also increases, so we had a lot of such repertoires, it could not fit into memory anymore. So what we need to do was to completely refactor how the data is stored. But because we had like a method that was called get sequences from other that was used from other classes, we could refactor the way the data is stored without changing any of the code that was actually using the repertoire class. So that was one example where we were able to completely switch to like from in-memory objects to lazy loading from optimized representations, and it greatly improved the speed. One other thing related to refactor, so one other way that the platform grows is not just to refactor, but also through extending with new functionalities. So even after everything is there and you think that is it is never it. So I guess no software is ever really done. And then there will always be some new functionality to add. In our case, it was, for example, we could see that new ways of representing the data would always be found, and then we would always need to kind of be able to extend the platform in that way. And also, for example, as new machine learning methods for our specific use cases were developed, we needed to be able to add them into the platform. So for most common cases, it's important to define how to extend the platform. And in our case, we did that with by defining the interfaces, or more specifically in Python, abstract classes, and then defining kind of a set of rules that these new representations would have to follow, and then new components were possible to add without changing anything else in the system, just adding a new component. One additional point is to that providing a full analysis specification was incredibly useful. The more complicated the analysis, the more important it is to provide convenient and transparent analysis specification. So initially we did this with just defining it in a script, in the code, calling the classes that we needed. But it turned out to be a bit cumbersome, especially as the number of options grew. So what we did in the end is that we switched to YAML specification files, where we define all the parameters that are needed for the analysis. And it also enables that the reproducibility of the analysis, because all the parameters are explicitly specified there. It's also related to keeping track of how each result was produced. So we have this on the level of the full analysis, but also on different levels throughout the code. So for intermediate results and so on. One other point I wanted to make was to follow the standards. Because no matter how good the platform is, it will always need to work with the existing software that does something else, or the next step in the analysis and so on. And to make something like that possible, having established standards in terms of storing of data formats and also software standards is very useful. For example, in immuneML, we follow the standards by the adaptive immune receptor repertoire community, defined for repertoires how are they supposed to be stored, but also some general software standards that we'll mention more about later. One other point is that also if you produce in some intermediate results in the analysis, they should also be in some standardized format for example, post-analysis with some other tool. One other point is to try to explicitly model the workflow in the code. That is also something, for example, how some tools like even Apache Spark works, where you have separate definition of what needs to be done, and it's separated from how it is actually done. So by having the definition of how things are supposed to be done, first it makes it readable, and second it decouples the implementation from the definition of what needs to be done. So one step in the workflow, in the analysis, could be easily replaced or optimized without the need to change anything, and it's really clear what is happening. So, for example, we had this trained machine learning model, which is a specific implementation of one instruction, applied machine learning model, which was another instruction, another workflow that was defined in this way. And the final point from my side before I let Lonneke do some talking is to make your code publicly available if you're developing a platform. It makes sure that it shows trustworthiness from your side, but also enables transparency. Assumptions are much easier to look into. And it also helps with the reproducibility. And for more on that, you can see the paper by Gachetel Santve and colleagues from 2013. And we'll reference all the papers I mentioned in the end. So with that, I let Lonneke do the talking.
Thank you very much. So Milena just talked a bit about more the technical aspects of developing a platform for bioinformatics, and I will talk a bit more about the usability side of things. So first of all, no matter how good your code is and how perfectly it has been developed, it would be completely useless if it is not actually accessible to people. So to ensure accessibility of your platform, the most important thing is, of course, that it should be easy to install, which might seem a bit too obvious, but it's definitely good to get some colleagues to test out if your code is actually able to be installed easily. Because one thing that we ran into when developing in immuneML is that about half a year ago, we had planned a day that we wanted to get feedback from our colleagues and lab mates on immuneML usability. And we actually found out that nobody was able to install immuneML at that point because of some dependencies that we had in the code for making plots through R, which actually turned out to be really difficult to install for people. Which also brings me to the next point: be aware of dependency issues. And if you have any dependencies in your code that people often struggle with installing, it is definitely worth the time to just go through it and replace them with something else, if that is possible at all. So in our case, we replaced all the R functionalities with Python functionalities, which was a lot smoother to install. If that's not possible, you can also consider if maybe some of your dependencies can be made optional. So in the case of some more difficult to install machine learning methods that only would be used by certain people in certain use cases, we've made them optional so that they can just be included as an extra during installation if desired. Another way to deal with difficult dependencies is to provide a Docker image. So a Docker image is basically a containerized version of your code that is executable and that already contains all the environment setup and dependencies inside it. And of course, you can also omit the need for installation altogether if you have a web interface. So there are command line interfaces and there are graphical interfaces. And for bioinformatics, I would say that having a good and usable command line interface is definitely a must, because command line interfaces are very useful if you want to create automated pipelines and so on. But it can be also relevant to consider if you would, in addition, need a graphical user interface. And for immuneML, we decided that we did want this. So we have a Galaxy web interface. Maybe some of you are familiar with Galaxy. So Galaxy is used for a lot of, as a front end for a lot of bioinformatics tools. So in this Galaxy interface, on the left side, you see the tool menu. So here, different immuneML functionalities are made available as so-called Galaxy tools. And then the results of these tools, they appear on the right side in this green area, which is called the Galaxy history. So those could be the output files from your analyses. And then, of course, these history elements can then also, again, be used as the input to the next Galaxy tool. And that way you can create a whole workflow within this Galaxy web interface. And the advantage of these history elements is that you can very easily also rerun them and tweak the parameters a little bit or rerun them on a different data set. And you can also share these history elements with other people or the completed history. So that way you can have a collaborative analysis on the same data set between different people. And if you are designing interfaces for your users, it's useful to think a bit about what different user groups you expect. So in the case of immuneML, we identified two main user groups that are a bit distinct. So on the one side, we have the immunologists, and on the other side, we have bioinformaticians. And they both have a bit of a different background and different desires and different ways of interacting with immuneML. So the immunologists, they are very familiar with their own data set, but they don't know so much necessarily about machine learning. So they need a little bit of explanation as to the different machine learning terminologies. So you need to put in a bit of extra effort to connect the different machine learning concepts to the biological concepts that they are already familiar with. And they also need a bit more guidance when it comes to making machine learning decisions. So they might not want to be able to change every parameter without limits, but rather have some a bit more standardized workflows that are generally sensible and have some ability to change parameters within that, but not have access to necessarily all the functionalities inside immuneML to not overwhelm them. And on the other hand, the bioinformaticians, they want full control over the parameters, and they want to easily be able to switch between this Galaxy interface and the command line interface. So for one Galaxy tool for training machine learning models, we made two different versions aimed at the immunologists and bioinformaticians. And in the case for the immunologist interface, we have a bit more extensive help messages where also the machine learning concepts are worded a bit differently or explained a bit more. And we have some more guided selection for options. So we don't expect them to write this YAML specification that Milena showed earlier, but we, so for example, if it comes to data representation, we don't want them to directly choose, like, I want to use this or that representation, but rather we ask some questions about assumptions that they have about their data and how a possible disease signal would look like in their data. And then based on that, we infer what would be the most appropriate data representation for them. And the bioinformatician, on the other hand, they just have a field where they can upload the same YAML specification that they would use on the command line. So that way they can be developing a use case locally on the command line, and then if they want to share it with a collaborator and or collaborate on the process on the different steps, then they can just upload this same YAML specification to the Galaxy interface and run it. So if you are interested in providing a Galaxy web interface for your own project, then there's some pros and cons to consider. So on the positive side, the Galaxy web interface, or any kind of web interface for that matter, really improves the accessibility. And this was also really the main reason that we for immuneML chose to have this Galaxy web interface, because we wanted to lower the bar as much as possible for people to get started using immuneML. We want people to just be able to go to a website and click some buttons and get some initial results out. as an advantage of GUIs in general over command line interfaces is that it gives you a bit more abilities to create a more intuitive interface, like the ones that we made for the immunologists. And specifically for Galaxy, it enhances reproducibility and collaboration because of these Galaxy histories. But then, of course, everything comes at a cost. And there's a lot of time and effort that was spent to develop this Galaxy Web Interface for immuneML. I think it was definitely about a year of active development until it was in the state where it is now. And it is also not easy necessarily, so you need some Galaxy experts to help you out. So we also had a team of people more specialized in Galaxy working on this. And of course, if you are going to provide a web interface where people want to upload their data and run computations, you need to be able to have the resources for that. So you need to have a place where people can store that data. You need to have a server where people can run the computations. So another thing to consider when you produce your output, it might be there might be a lot of outputs that are produced. At least in the case of immuneML, this is a complex platform. There's a lot happening. There's a lot of intermediate steps with intermediate results that are being stored. And it's really nice to be complete, but disadvantage of that is that the user can be overwhelmed with the amount of folders that are produced and how to even navigate them and where to find the results that I'm most interested in. So a solution that we came up with for immuneML is that we are automatically producing these HTML pages, which are used to navigate these results folders, so that you can just click through the different pages and find the relevant plots or other kind of results that were produced. And in this HTML page, we also link to the YAML specification that was used, and to we write down the version of immuneML that was used and the details about the dataset that was used. So that way you have your input and your output all packaged together. So you don't have a situation where you just find a random output file somewhere and you have no idea how it was produced, with which version or what input dataset, but rather you have everything together. So that is really nice. Of course, you will also want to provide user documentation and tutorials. And depending on your platform and how complex it is, you will need varying degrees of documentation. So I can't really tell that how much you exactly need. But I at least identified 3 main components that I think are important in our documentation. So those are the quick start tutorial, installation guide, and then other types of usage tutorials. And I will tell a little bit more about specifically the quick start tutorial, because I would say that this is the most important tutorial that you can have, because this is kind of your first impression that you give to the user. This is where you want to lure the user into starting to use your platform. So you want to have well, it's in the name, it's a quick start. So you want to aim to have it as quickly as possible, that the user can run it in at most 5 minutes, maybe 10 minutes. At least in the immuneML case, it is closer to 10 minutes, and we hoped it to be closer to 5. But that way, you don't want your user to get bored. You just want them to quickly go through it. And if you are a little bit cynical, you can also think that the user might even want to just test if your platform works at all, because there's a lot of software out there online, and maybe some of it is not well maintained. So you just want to convince your user that actually your software is working and it's good. And also for the quick start tutorial, you can think again about the different user groups that you have. So in the case of immuneML, we ended up with 3 different quick start tutorials. So we have one for the immunologist on the Galaxy interface, one for the bioinformatician on the Galaxy interface, and one for the bioinformatician on the command line interface. And in the quick start tutorial, you want to provide a small but meaningful example dataset. and then just go step by step through the essentials of the analysis. And if you notice that you're trying to pack too much into it, or that it's too much details and the tutorial ends up being too long, just separate that to a different tutorial and link to that instead from the Quick Start tutorial. So really just have the bare minimum essentials and have links to the more detailed tutorials in addition. And it's also important to not forget to give some kind of interpretation of the results that are produced, because you are very familiar with your platform and what it does and what the results look like. But for a user, it's just their first encounter. So it's not immediately obvious to them when they see a plot what it means or where they should be looking. It is really nice if you can add one paragraph on how to interpret the main results that came out. And also don't leave the user hanging in the end, but just give them some pointers on what they can do next. Because if they made it all the way through their Quick Start tutorial, they might actually be very interested and they might want to start using it on their own data and see what else your platform has to offer. So you want to have some useful pointers to other tutorials or things that they can do. And with the Quick Start tutorial, it's also important to get all the feedback that you can, because this is such a, because this is your first impression. Just ask all of your colleagues and collaborators to just try it out, and it shouldn't take them too much time if it's a 5-minute tutorial, because you definitely don't want people to get stuck during the Quick Start. You want everyone to be able to do it. And if you have a graphical user interface in particular, it can be nice to also have some video tutorials, because it is sometimes not so easy to explain where to click and all these things. So for Galaxy, we have, for example, the Quick Start tutorial also as a video form, as well as some other tutorials on how to use immuneML. But no matter how much documentation and explanations and tutorial videos you provide, users will always find very creative ways of using your platform in a way that you had not intended. So you also have to think a little bit about what happens when things go wrong. What does the user meet when they fill in the wrong input, for example? So aim to have understandable and specific error messages, at least for the most common mistakes. So everywhere where the user can fill in some input, if it's a number, check if it is, for example, if you're not allowed to have a number below 0, just make sure that you have these checks and that the user is not met with some generic Python error in this case, but that they instead get some pointer as to what they have to change. So there's not one quick way of having understandable error messages. It's hard work. It is just iteratively going through it, testing out your platform a lot, see where you maybe make mistakes yourself, like if there's something wrongly formatted in the input, for example. But it's definitely worth doing, because this is also, in a way, a form of documentation. There's a lot of users that might not bother really reading through all the documentation that you provide, and that instead just read the errors that they see on the screen when they give the wrong input. Also, you should either crash early or crash not at all. But the one scenario that you really want to avoid at all costs is if your tool has been running for a very long time, maybe for hours, maybe for days, that it crashes somehow because you were creating a plot all the way in the end, and then all of a sudden all of your results are lost. So that's definitely what you want to avoid. So you want to sanity check all the parameters that the user has specified before starting to run the main analyses. So in the case of immuneML, what we did is for all the components that the user can choose, like the different data representations and machine learning methods and reports, we have this build object method where we just use assert statements to make sure that the input is correct, and then create an object if the input parameters are valid. And if there are any less essential components of your analysis, like in our case, you have you can train a machine learning model, but in addition specify some report that plots some characteristics about your data. But if there's anything that goes wrong in these reports, we don't crash, but we just catch those errors and redirect them to a log file. So that can also be useful to consider. All right, so if you found this presentation interesting and useful, and you are maybe working on your own bioinformatics platform, then here are some recommended pointers to other reading that you can do. And for anyone who is working with adaptive immune receptors, we definitely also recommend you to check out our manuscript, which is on the bioRxiv for immuneML. And the website, of course, immuneML.uio.no. And then finally, I would like to thank all the colleagues and collaborators that made the immuneML project possible due to their contributions and feedback. So thank you very much.
Thanks for your great presentation. I believe this experience and the development process are really valuable for everyone who is trying to develop not just bioinformatic tools, but like also maybe other software. So for a question like if you have any question, you can just write it down. Okay, see one question. To what extent have you collaborated with Elixir or other groups with regards to the Galaxy backend?
Well, I would say a lot. So all the tools that we have were initially developed by people from Elixir in collaboration with the USIC people, actually. So a lot.
So any other question want to ask? And okay, there's one question from my side. Because like I'm not really a good software developer, but I'm always wondering, like, when you develop such a big software, do you design this abstract or like blueprint for like structure of different components from the very beginning, or do those structural components are kind of slowly built up during like the different iterations you mentioned?
So I think what we did was start with something that was structured, but not the final version, and then improve it over time. So you should, if you want to make a big platform, you should think about it from the start. But of course, what you do initially will not be what you end up with. But it's good if it's a good starting point, I would say.
I think it's kind of a combination of the two options that you mentioned also, because I would definitely say the very core, I joined the immuneML project later. I call you immuneML. Milena started it, but I joined in later. And when I joined, there was definitely already a core structure there that has not changed too much. But there are some quite essential components that have been added or altered a lot during the time that I joined also.
Okay, and there's some scientific question about immuneML. So what kind of data set do we need to use immuneML? Is it CDR3 or completely sequences or other kind of metadata?
So I would say that we mostly work with these CDR3s. There's some possibility for us to it might be possible in the near future to also use complete receptor sequences, because we have thought a bit about that. But we mostly work with CDR3 sequences or repertoires of CDR3 sequences. And both, we have a single chain, but also paired chain data.
Yeah, for both B and T cells.
Yeah.
And okay, there's another question. So, have you used immuneML with antibody sequences as well as TCRs? So it seems like the comments says it seems like more TCR centric for now.
No. So we did we had 3 use cases to show the usability in the manuscript, 2 of which were on TCR, 1 was on BCR data. So you can use immuneML with either one.
It can be used with either, but they're essentially treated the same way.
Yeah.
So we don't have any specific antibody things that wouldn't work on TCRs, but all the functionalities that we have, that they can be applied to both.
Okay. So is there any other question? Okay. So okay. There's another one. How useful is it? Is beta strand only TCR datasets compared to paired alpha beta?
That is an interesting question, because that was a project that I was working a bit on specifically. I don't have a very conclusive answer, but it seems that at least the beta chain is well, I think it can depend a lot on different studies and what you're exactly looking for and so on. But it seems that in a lot of cases, having beta chain only is already very useful, and especially if you consider the fact that in order to have paired chain data, you often don't have as high throughput sequencing. So it might be that having a bigger beta chain only, like a bulk sequencing beta chain data set, might be more useful than a smaller pair chain data set. But it's not like there's one valid answer necessarily to that question. I hope to give you more detailed answers on that in a couple months.
Okay, so is there any other question or question I have missed? Okay, so okay, and in the meantime, like if people want to give a talk like this, you can join our Slack channel, and also provide you can sign up for a speaker and maybe suggest speaker using the form we have. Okay, so if there's okay, there's another question, I believe. So, how have you obtained your test bed data from open content aggregators, which is the largest data size you have analyzed so far?
We've used previously published data sets, like for example, this Emerson data set that was nearly 800 repertoires in total, but the largest data set that we have worked with is in one of the use cases in the immuneML manuscript, where we simulated data using this program called Olga, and that was 2,000 repertoires of 100,000 sequences each.
Okay, so is there any other question? Okay, if not, then thank you so much for your valuable presentation. And I think that's all for the talk today. And looking forward to see other researchers presenting their talk here in this platform. Thank you so much.